So you guys were ready to start about 2 minutes ago. I turned the lights down, and all of a sudden everybody got quiet. Kind of odd. So the run to Thanksgiving is almost here. How was the exam on Friday? Comments? Nobody has any comments? Yes, Omar?
I think that I was talking about a friend about this, and like it seems like the test kind of didn't really cover the full breadth. Like it felt like it covered very specifically some topics, but ignoring others. Like for example, I noticed that like it seemed like it was like a couple of questions about blood clotting, and then like a couple of questions about elastase. So it wasn't like a kind of. I felt like you were testing like very specific parts of it, but kind of.
Okay, so he felt that there was some lack of coverage of some things. What did you feel like should have been covered?
Well, like for example, ATCase was like on the test.
Mm-hmm.
I feel like that's one thing that like was talked about. We talked about quite a bit.
Uh-huh.
It's kind of not there.
Okay. Okay.
I didn't really feel like it impacted my ability to take the test, but I felt like
Okay.
the test is kind of narrow.
Okay.
Focused.
The test was narrow in focus. All right. I see a couple nods for that. Other comments? Yeah. This test was shorter. Okay.
It seems like we had a little bit extra time that just span on some of these reflections.
In fact, this test actually had one more question than the last one did. So if it seemed shorter, but it wasn't a shorter test. Yeah. Which is good. That's good. But you felt like you had more time. Is that in general? People felt like they had more time? Okay. I was a little concerned because I thought people seemed to be staying a little bit longer, so maybe my impressions weren't what your impressions were. Okay. Other comments? Yeah? Expecting more questions on glycolysis. Okay. So it was kind of a tough test to write because it's a fairly broad set of topics to cover. And that's why I think that the comments about, it seemed like it was places of things missing or other things that there weren't that much of and so forth, probably are true because there's just so many darn different things to cover on that exam. I hear you. I think one of the things students always expect to see is that I'm going to ask them to regurgitate a whole pathway, write out the pathway, and that's generally not going to happen, all right? I'm going to test things of a pathway or ask you things of a pathway that you need to know, but I'm not going to ask you to come back and just regurgitate a pathway for me. So that's one thing you won't see from me in general. Okay. Well, appreciate the feedback. The exams are being graded. I have given the TAs the hopeful incentive that they'll be graded by the time Thanksgiving arrives. So I'm hopeful. I don't know. I can't promise anything. I'm hopeful they will be available by Wednesday sometime. And they are working very hard on that, and hopefully we'll get to that point. Okay. Well, let's move ahead with new material. I will say one more thing, and that is on the final exam, you will get to use a note card full of all the things you can squeeze onto a single note card. The rule is you have to get the note card from me. No ifs, ands, ors, or buts. And, this is the odd part, you have to get a note card from me. Okay? So if you come to the exam and you don't have a note card from me, you're going to lose points. Okay? Even if you don't want to use one, that's fine. You can bring a blank one, but you have to get a note card from me and bring it to the exam. Does that make sense? I will bring note cards next week sometime, and then you can get the note cards. But it's essential that you get the note card from me. If you bring in a different note card or you don't bring in a note card at all, you'll lose points. Okay? Most people don't complain about having a note card. It's a big note card, too, by the way. Yeah. All right. Positive rumble in the class with that news. Okay, so last time I started talking about regulation. And as I said, I'm going to talk about regulation, and then we will talk about regulation after I finish gluconeogenesis, because I think you will start to see regulation in a different light after that point. Okay? So this pathway, or this scheme on the screen shows the three enzymes that are regulated in glycolysis. They are hexokinase, which is the first one, PFK, which is the third one, and pyruvate kinase, which is the last one. Okay? As I said, the one that's the most important for our purposes is PFK, and we'll talk later about why an enzyme in the middle of the pathway is important. Hexokinase I don't really talk much about. It's regulated by what's called substrate level regulation, and to be honest, it's not the most important type of regulation for us to worry about. So I don't see any reason to spend time on it. PFK is regulated, however, allosterically, and it's regulated by several things. And so understanding that is important. And the last one, pyruvate kinase, turns out to be a very interesting enzyme for us because of one of the things that it's regulated by. So let's take a look at pyruvate kinase. Pyruvate kinase is regulated both allosterically, by the things that you can see on the bottom, and by covalent modification. Okay? So pyruvate kinase, when it is phosphorylated, it is less active. When it's dephosphorylated, it's more active. What does the phosphorylation? Well, there's our friend, protein kinase A. Okay? Protein kinase A is involved, as we shall see, in phosphorylation of many proteins, and this is one. Okay? Now, for our purposes right now, the more important consideration is actually right down here at the bottom. Fructose 1,6-bisphosphate is an allosteric activator of that enzyme. All right? An allosteric activator of the enzyme means it's going to turn the enzyme on. In addition, fructose 1,6-bisphosphate is an intermediate in glycolysis. It's an intermediate in glycolysis, and if we think about where it appears in glycolysis, it gets kind of interesting. Okay? Fructose 1,6-bisphosphate in glycolysis is converted into glyceraldehyde 3-phosphate and DHAP by the enzyme aldolase. What was the noteworthy feature we had about aldolase on the overall scheme? High positive delta G 0 prime. And we said in order for the reaction to proceed, the cell had to do what I called pushing, which was increasing the amount of substrate, and pulling, which was removing the product. Okay? This helps to remove the product. So if we think about what's happening in glycolysis, let's say that the cell is all of a sudden needing a whole bunch of energy. So the body dumps a bunch of glucose into the cell. And the glucose goes to glucose 6-phosphate because of hexokinase. And it goes to fructose 6-phosphate. And it goes to fructose 1,6-bisphosphate because PFK has been turned on. But then it hits the roadblock. And what's the roadblock? Aldolase. The roadblock is aldolase because with a high delta G 0 prime, there's not a lot of it that's going forward. So what happens is the cell is dumping, and fructose 1,6-bisphosphate concentrations are increasing. And when they start increasing, guess what they interact with? Pyruvate kinase. And pyruvate kinase starts metabolizing PEP, which means PEP concentrations fall, which means that 2PG concentrations fall, which means that 3PG concentrations fall, which means that 1,3-BPG concentrations fall, which means that glyceraldehyde 3-phosphate concentrations fall, which means that dihydroxyacetone phosphate concentrations fall. You get the picture? We've started pulling away the products of the aldolase reaction. Yes, sir?
Aren't you pulling away the reactants as well?
The reactants of what?
Of the aldolase.
No, because the reactant of the aldolase is the fructose 1,6-bisphosphate.
Right, you're pulling it away for this reaction.
Well, this is a very tiny amount that's binding the enzyme. So, no, this has no impact on the concentration of fructose 1,6-bisphosphate. Remember, enzymes are present in way, way lower abundance than substrates are. Okay? So, pushing and pulling helps this reaction to go forwards. And this setup here is facilitating the pulling. Make sense? Clear as mud? Okay. Is there a question? Okay. All right. So that is what the sum of what I'm going to say about regulation at this point. I'll say some more when I get down here to the bottom after I do gluconeogenesis. But before I do gluconeogenesis, I want to mention just a word about gluts. We talked about gluts before. when I talked about the insulin receptor. And the insulin receptor was stimulating the movement of GLUT4 to the surface of cells. Okay? And GLUT4 is, as all the other GLUTs are, is a membrane protein that facilitates the movement of glucose into cells. Okay? GLUTs turn out to be really important because, as we will see later next term when we talk about membranes, membranes are essentially impermeable to almost every biological molecule. The only way they get across membranes is for specialized proteins to let them in. GLUTs are therefore very important for cells to have food in the form of glucose. Well, that actually brings up a little sideline that I want to talk about here relative to cancer. Okay? Cancer cells are, as we know, undifferentiated. They are typically growing more rapidly than our regular cells. And as such, they have needs that are different from regular cells. Okay? If they're metabolizing more rapidly, if they're dividing more rapidly, they have more energy needs. And when we think about energy needs inside of cells, one of the things we think about is oxygen. Okay? Oxygen is important because, as we'll see next term, oxygen helps the cells to make the maximum amount of ATP that they can for a given molecule of glucose. One of the things I didn't mention, but I'll mention right here, is if I go through glycolysis, I go all the way through glycolysis, and I stop at the end, okay, I have made 2 pyruvates, and I've made a net amount of 2 ATPs. That's the sum of it. Okay? If I don't have oxygen, then to keep glycolysis going, I've got to convert that pyruvate to lactate. That takes up the NADH that I made. So my overall net product under fermentation conditions is a grand sum of 2 ATPs. That's what I get. Okay? On the other hand, if I have oxygen present, and that pyruvate can be converted into ultimately CO2 by the citric acid cycle, the amount of ATPs I can make is 38. 38 ATPs in the presence of oxygen compared to 2 ATPs in the absence of oxygen. That's a lot. If I want to lose weight, do I want to go aerobic or anaerobic? Everybody says aerobic. They're completely wrong. You want to go anaerobic. What most people think of as aerobic is actually anaerobic, folks. Because you're using oxygen faster than your body can deliver it. That's why you're running, and that is why you're breathing heavily. And the more glucose you burn per fewer ATPs, the more glucose you got to keep burning, right? So when you hear, next time you hear your friend talking about, oh, I'm going to go get aerobic at the gym, okay? You say, well, I listened to Ahern, and he says you're going to go anaerobic, right? You're going to lose more weight if you go anaerobic than if you go aerobic. And the only way you're going to go anaerobic is if you get your butt out and exercise. Okay? All right. Now, why does that all tie into this? Well, tumor cells have greater energy needs, and tumor cells, when they start out, don't have a good supply of blood. Tumor cells turn out to produce hormones that stimulate the growth of blood vessels into them. There's a hormone called angiogenin that stimulates the growth of blood vessels. Okay? And even there, you can look at this sort of this tissue that's here. You see the little blood vessels growing there, but we can imagine there's still pockets in there that aren't getting very good blood supply. Tumor cells are what we call hypoxic. They're needing oxygen. They're low in oxygen much of the time. Some people, in fact, think the hypoxia of tumor cells is a strategy for killing them. Okay? Well, hypoxia has many effects, right? one of the effects is one that's already built into our cells. Our cells have a way of responding to hypoxic conditions. It's by the production of a transcription factor known as HIF, Hypoxic Induction Factor, Hypoxia Induction Factor. HIF1, okay, is a transcription factor. And what it does is it can activate the transcription of other genes. Under conditions of hypoxia, HIF1 is made. So we have cells, and this is true of normal cells as well as cancer cells. We have cells in our body that are low in oxygen. They produce HIF1, and HIF1 turns on the production of some really interesting genes. Look at what HIF1 turns on. GLUTs. Surprise, surprise. If we are only getting 2 ATPs out per glucose, doesn't this mean we're going to need a heck of a lot more glucose? You betcha. All right? Hexokinase, first enzyme of glycolysis. Phosphofructokinase, the most important regulated enzyme in glycolysis. Aldolase, the one that has the energy hump. Glyceraldehyde 3-phosphate dehydrogenase, the one that catalyzes the only oxidation in glycolysis. Phosphoglycerate kinase, which produces ATP. Okay? Enolase, which makes PEP, and pyruvate kinase, which makes pyruvate. Okay? This is an all-star lineup of enzymes that HIF1 is inducing. And no, you're not going to memorize these enzymes. Okay? But it's an interesting set of enzymes. We can say, wow, cells that are low in oxygen have a response and a way of dealing with that by making it possible for the cells to metabolize more glucose. If you only get 1/19th as much ATP per glucose when you have no oxygen, you better have more of these enzymes abundant for cells to handle 19 times more oxygen. Pretty cool. Questions about that? Excuse me. Okay. So, that's pretty cool. Let's now turn our attention. We've finished glycolysis. Let's turn our attention to the synthesis of glucose, which is equally important, if not more important. All right? Now, one of the things that students get confused about is that they want to have all these pathways going on in all of the cells of the body, and they're not. Glycolysis goes on in essentially every cell of the body. In fact, it does go on in every cell of the body. But gluconeogenesis, the synthesis of glucose, does not. It occurs in specialized cells of the body. The primary place we will find gluconeogenesis occurring is in the liver. There are also some cells in the kidney and the gonads that can make glucose. But the liver is the primary organ for making glucose. Muscles don't make glucose. Muscles are completely dependent upon glucose from other parts in the body. Your brain is completely dependent upon glucose from other parts of the body. Your eyes are completely dependent. You get the idea, right? The way we're getting this is we're pulling this glucose out of the bloodstream, whether it's coming from our diet or it's coming from being made in the liver. Okay. Well, forget the thing about glycerol. Let's talk about gluconeogenesis. Here's the pathway that you saw before for glycolysis, only it's been modified to be for gluconeogenesis. With gluconeogenesis, I like to think of it as starting at the bottom and moving up. If you look at the pathway, all the enzymes that are colored in blue are the same enzymes as glycolysis, meaning that the reaction is simply run in the reverse direction. There are 10 enzymes in glycolysis, and 7 of the enzymes of glycolysis are the same in gluconeogenesis. Three enzymes of glycolysis are replaced by 4 enzymes of gluconeogenesis. The first one, which is the last one in the direction of glycolysis, the first one, pyruvate kinase, is replaced by 2 different reactions. And if we think about it, that makes a lot of sense because that pyruvate kinase catalyzed the Big Bang, right? And the Big Bang had so much energy going in the direction of glycolysis that that's essentially going to be an impossible barrier to get around by simply reversing the pathway. So the cell doesn't even try. Instead, the cell does what I call a two-step. Two separate reactions, each of which requiring triphosphate to go from pyruvate back to PEP. The first of these enzymes is one of the two enzymes of gluconeogenesis that's found away from the cytoplasm. All of the enzymes of glycolysis are found in the cytoplasm. 9 of the 11 enzymes in gluconeogenesis are found in the cytoplasm. 2 are not. The very first one and the very last one. The first one is an enzyme called pyruvate carboxylase. This enzyme is found in the mitochondria, and it converts pyruvate into a molecule called oxaloacetate. You can call oxaloacetate OAA if you want. Okay? OAA. Okay? This enzyme catalyzes a reaction that requires ATP. Let's see if I've got it up here. I think I do. Yes. Okay. Here's the reaction that it catalyzes. Pyruvate plus carbon dioxide plus ATP plus water gives us oxaloacetate. You'll hear a lot about oxaloacetate next term. It's an intermediate in the citric acid cycle. It's also readily made from aspartic acid. It appears in other metabolic pathways as well. Okay? This reaction, okay, requires a triphosphate and gets us half of the way to making PEP. This enzyme also, people have asked me a couple times during the term about coenzymes. Coenzymes being important. This enzyme has a very important coenzyme that you'll see many times in biochemistry. The coenzyme is known as biotin, B-I-O-T-I-N, and yes, that is an essential nutrient. It's vitamin B6, I think. Don't quote me on that. I think that's right. Okay. Biotin has the function of grabbing a hold of a CO2 for reactions that involve carboxylation, and that's exactly what's happening here. The name of the enzyme, carboxylase, tells us this is a carboxylation. And almost any time you have a carboxylation occurring inside of cells, biotin is a coenzyme involved in those reactions. Okay. There's biotin. Biotin's actually attached to a lysine side chain inside of the enzyme. It's physically attached to it. And this is what it looks like when it's grabbed a hold of a CO2. And no, you don't have to draw that structure. Okay. All right. The second enzyme of gluconeogenesis that's different is the very next enzyme. It's called PEP carboxykinase, or PEP-CK. It catalyzes the conversion of oxaloacetate into PEP. Notice that the energy source for this is not ATP, but instead is GTP. GTP has exactly the same energy as ATP does. Some enzymes use GTP. Most enzymes use ATP. This is one that uses GTP. The enzyme is called PEP carboxykinase. You can call it PEP-CK. Okay? So what have we done? We've taken two steps. We went from pyruvate to oxaloacetate. We went from oxaloacetate to PEP. So we're now back at the PEP intermediate. And we've used two triphosphates just for that one conversion, right? Remember, if we're going to make glucose, we've got to double all these reactions. So in essence, if we're making glucose, we've already had to use 4 triphosphates to get back to 2 PEPs. Now, at this point, I'd like to point out why it's really useful and why it's very important that pyruvate kinase is regulated. Remember I said it's really odd. Here's the last enzyme of a reaction that's being regulated. Feedback inhibition works really well for the first enzyme. Why in the heck do we regulate the last enzyme? This is critical, folks, because we've just blown 2 triphosphates to get back to 1 PEP. And what happens in the presence of pyruvate kinase if PEP is present? The reaction goes back to pyruvate, right? So what would I be doing if I couldn't turn that enzyme off? I'm trying to make glucose, I make PEP, but instead of going up the chain, I go right back down to pyruvate. That Big Bang has to be controlled. If you don't control the Big Bang, you do something we refer to as a futile cycle. F-U-T-I-L-E. A futile cycle occurs when we make something and break it down, and make something and break it down, and make something and break it down, and we don't get anything out of that except for heat. And meanwhile, we're going to lose a lot of energy. Okay. Well, the good news now is if we go back to our overview, is we just now, to get way back up here, all we have to do is run the reactions backwards. Same enzymes, all right? We're now pushing this up from the bottom, moving upwards, up, 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 until we get to fructose 1,6-bisphosphate. Fructose 1,6-bisphosphate is converted to fructose 6-phosphate, all right? And you'll notice we use a different enzyme. Why do we use a different enzyme? Well, if you think back, you'll remember I told you that PFK catalyzed a reaction that was energetically favorable in the glycolysis direction. If we try to fight that energy thing, we're going to lose the battle again, folks. We're going to lose the battle. So rather than do that, we change the enzyme, and we can't just change the enzyme, we have to change the reaction itself. So, in the direction of glycolysis, ATP is being converted into ADP, right? Going from fructose 6-phosphate to fructose 1,6-bisphosphate required an ATP. If we were to simply reverse that reaction, we would be making ATP again, right? This doesn't make any sense. We're making glucose when the cell has plenty of energy. That's the only time we make glucose, when the cell has plenty of energy. Liver cells in particular, right? They got plenty of energy. They've got plenty of ATP. And by the way, liver is full of ATP because its blood is fairly rich in oxygen. Okay? If we have plenty of ATP, and we try to make a reaction that makes more ATP, and ADP is limiting, that reaction isn't going to go. So it doesn't make sense to just simply reverse the reaction from 2 perspectives. One perspective being energy, and the other perspective being we got plenty of ATP, which is also energy. All right? So instead what the cell does is it catalyzes a reaction that involves the clipping off of a phosphate. It pulls the phosphate off of position 1, and in doing so, this reaction becomes energetically favorable in the upwards direction. Why? Because remember that putting that phosphate on took energy. If I simply clip the phosphate off, I release energy. Now the reaction is favorable energetically going upwards. The enzyme that catalyzes this is called fructose 1,6-bisphosphatase. You can call it F16BPase. And here the names are going to start running into each other, so be careful. An A is at the end means it's an enzyme. So F16BPase is an enzyme. F16BP is a molecule. All right? We've now got fructose 6-phosphate. Well, next enzyme is glycolysis enzyme again, and then we get to glucose 6-phosphate, and exactly the same considerations we had for this enzyme, the fructose 1,6-bisphosphatase we have for the next enzyme. We don't want to run it backwards simply. Instead, we clip a phosphate off, makes it energetically favorable, and this enzyme is called glucose 6-phosphatase, or G6Pase. And this enzyme is found only in the endoplasmic reticulum. Only in the endoplasmic reticulum. And only in the endoplasmic reticulum of liver, kidney cells, and gonad cells. We've made glucose. Now, for everything down here up to that point, we've got 2 of everything, so we've got to keep that in mind. Okay? If we do that, what do we produce? Well, it took 2 ATPs, it took 2 GTPs. Here's 2 more ATPs here it's going to take. All right? So there's a total of 6 triphosphates it's making, and we use no more ATPs going up here, but we produce no more either. It takes 6 triphosphates to make glucose from pyruvate. 6 ATPs to produce glucose from pyruvate. If we go through glycolysis, we had a net of 2. Okay? Glucose gave us 2. Synthesis of glucose costs us 6. It tells us something very important. We do not want to have glycolysis going on at the same time as we have gluconeogenesis going on. Because why? We're going to be running a futile cycle, burning more glucose than we make, burning more ATP than we make, and we're not going to produce anything but heat. Okay? This turns out to be a major consideration for the regulation of glycolysis and gluconeogenesis. Generating some heat right now in here might not be a bad idea, might it? Okay. So let's think about the regulation of these 2 pathways. The regulation is something we call reciprocal. And reciprocal regulation turns out to be a very, very important and useful concept for us to understand with respect to metabolism. Reciprocal regulation. In simple terms, what it means is that things that turn off catabolic pathways turn on anabolic pathways, and vice versa. We want one to be on when the other is off. We don't want them both running at the same time in the same cell. Notice I said in the same cell. Because our liver is making glucose at a time when our muscles are using glucose. So it can happen in different parts of our body at the same time. But we don't want them both going on at the same cell at the same time. And this is important because if you think about it, the enzymes are all intermixed in the cytoplasm for the most part. They're all intermixed in the cytoplasm. Let's look at the regulation. Here are the enzymes that are different. You notice that we don't distinguish glucose 6-phosphatase from hexokinase. So the only 2 we'll consider are these 2 sets here. Let's look at the first of these, PFK. Okay? PFK is activated by fructose 2,6-bisphosphate. Fructose 1,6-bisphosphatase is inhibited by fructose 2,6-bisphosphate. What's turning one on is turning the other one off. PFK is turned on by AMP. Fructose 1,6-bisphosphate is turned off by it. On the other hand, PFK is turned off by citrate, but fructose 1,6-bisphosphatase is turned on by it. Citrate is an indicator of high energy inside of a cell. High energy means we don't want to be burning any more glucose, like lighting our fireplace in the summertime. We don't want to do that, right? Instead, we want to save this energy, store it in the form of glucose. Now, you see it's not perfect. There are some things that affect this enzyme and that don't affect this enzyme. But nonetheless, it's striking how the two enzymes are differentially affected. Okay? Over here, right, we see, for example, that ADP turns off pyruvate carboxylase. It also turns off PEPCK. This upwards pathway is turned off by something that's indicating low energy. We don't want to be synthesizing glucose when we don't have any energy to do it, right? On the other hand, pyruvate kinase is turned off by ATP, meaning we have high energy. We don't want to be breaking down, making more pyruvate. Because if we have plenty of ATP, we want to be moving in the upwards direction. As noted, fructose 1,6-bisphosphatase activates pyruvate kinase, and that's important for pulling that aldolase reaction. Okay. Questions about this? Yes?
You said there was an indicator that says low energy. Which one was that?
He said, I said there's an indicator that indicates low energy. What was it? Well, there's a couple of them on here. One is ADP, indicates low energy, because you get ADP when you've burned ATP. Another indicator of low energy here is AMP. AMP is an indicator of very low energy, actually. Okay. Other questions? Cool stuff. All right. Well, I'm going to tell you a story about a very interesting enzyme. And what we're getting ready to talk about right now is actually related to the synthesis of fructose 2,6-bisphosphate. We haven't talked about where that came from yet. All right? So now we're going to talk about how it is that cells make fructose 2,6-bisphosphate. The enzyme is called phosphofructokinase 2, or you can call it PFK2. So it sounds like PFK, and it is similar to it, in that it's putting a second phosphate onto fructose 6-phosphate. In the case of PFK, it's putting it onto carbon number 1. In the case of PFK2, it's putting it onto carbon number 2. That's very simple. So PFK, you can also think of as PFK1 if you want to. PFK1 puts a phosphate onto carbon 1. PFK2 puts a phosphate onto carbon 2. Remember, though, that that fructose 2,6-bisphosphate is not an intermediate in glycolysis. It's simply using the fructose 6-phosphate from glycolysis. Okay? Something else turns out to be really interesting about this enzyme. Because in addition to putting a phosphate onto fructose 6-phosphate to make F26BP, PFK2 also has the ability to remove it. And I'm not talking about simply the reversing of the reaction. I'm talking about the physical removal of the phosphate by the clipping reaction that we saw before. It turns out that this enzyme is Jekyll and Hyde. This enzyme has the ability to use ATP to attach a phosphate onto position 1, and it has a phosphatase activity associated with it, with the ability to remove a phosphate from position 2. Jekyll and Hyde, folks. This is about the only Jekyll and Hyde enzyme I know in all of biology. This enzyme right here turns out to be really critical to control. You got to control one activity versus the other activity. Okay? Under some circumstances, we want to be making F26BP. In other circumstances, we want to be breaking down F26BP. And this one enzyme has the ability to do both. Because this enzyme has two activities, I give it two names describing which activity is active at any given time. So when I call it PFK2, the part that makes F26BP is active. When I call it F26BPAs, okay, I'm talking about its ability to remove a phosphate. Okay. Now, this is going to bring up the figure that's the most hated figure that I show in all of this class. I'm warning you before I click on it. Here we go. Now, if you take my words and convince yourself that this is a horrid figure, then you'll probably make that be the case. It's not as bad as it looks. All right? Here's our Jekyll and Hyde enzyme. Over here we see that the PFK2 is the active part, and over here we see that the FBPase 2. And by the way, you can also call it FBPase 2. I call it F26BPase, but you can call it FBPase 2 if you'd like. Okay? Over here we see that the FBPase 2 is active, and the PFK is inactive. What's the difference between them? It's very, very simple. The difference is a phosphate. You put a phosphate onto it, you favor the FBPase 2. You take a phosphate off of it, and you favor the PFK2. That's very simple. Everything else on there tells us how the phosphates get on and come off. Everything else on there tells us how the phosphates get on and come off. All right, so in studying this figure, start right here. Phosphate on versus phosphate off. Okay? If you don't know that part, you're not going to know any of the rest of it. All right? How does the phosphate get onto there? Oh, that's very simple. Our friend, protein kinase A, puts it on there. How does it come off of there? Oh, our friend, phosphoprotein phosphatase, takes it off of there. Epinephrine favors protein kinase A being active. And insulin favors phosphoprotein phosphatase being active. Okay? If if PFK2 is active, that means that fructose 2,6-bisphosphate is being made, right? And when fructose 2,6-bisphosphate is being made, what happens to glycolysis? Glycolysis is stimulated because PFK is active. If I have insulin, insulin, as we will see, stimulates phosphoprotein phosphatase being active. Okay? We favor this direction when insulin. We favor the direction to the right in the case of, I may have said that wrong earlier. Favor the direction to the right in the case of epinephrine, to the left in the case of insulin. Let's think about what that means. Insulin. When we have insulin present, what is happening to glucose? It's being brought into the cell. When glucose is a poison, right? It's a poison for cells too, right? Cells have to do something with that insulin, right? One of the things they can do with it is burn it up. And they do. We'll see the other thing they can do with it is they can turn it into glycogen. Okay? Under conditions of epinephrine, okay, cells are running out of glucose. The body is running out of glucose. It needs to make glucose. When it needs to make glucose, it does not want to have glycolysis going. It wants to have gluconeogenesis going. For gluconeogenesis to go, we want to be making, that is, we want to be breaking down F26BPAs, which is going to allow FBPase 2 to be active. Now, one of the things that's going to happen with this figure is you're going to see, just like I'm doing it in front of you, it's hard in one sitting to sit and digest this figure. I want you to take it home. I want you to write it out. I want you to think about it. And I want you to start exactly where I started, with what the two different types of the enzyme were, what made one active versus the other active. Because this enzyme is at the core of reciprocal regulation of glycolysis and gluconeogenesis. Understanding that is going to require an understanding of this. Because it tells us how F26BPase is made. I'm sorry, how F26BP is made. Now I'm doing it too. And how F26BP is broken down. Questions about this? Stunned silence. Yes, sir.
Between glycolysis and gluconeogenesis, is there a difference in how long it takes to go from one to the other?
So between glycolysis and gluconeogenesis, is there a difference in how long it takes to go from one to the other? Essentially, for a cell, no. Yeah. That switch is not something that a cell would need to do instantaneously. Because again, remember we're talking about liver cells, kidney cell, a few kidney cells, or gonads. All right? So for the most part, that isn't, those aren't cells that are typically going to have to make a quick decision. It's not like a muscle cell, because muscles aren't making glucose. Okay? Other questions? Okay, I've got a dandy song that finishes this all up, and I want you to sing this one loud. It's my favorite song of all the ones I've written. It's called Gluconeogenesis. When cells have lots of ATP and NADH2, they strive to store this energy as sugar, yes they do. Inside of mitochondria, they start with pyruvate, carboxylating it to make oxaloacetate. Oh, gluconeogenesis is so exhilarating. Memorizing it can really be exasperating. Liver cells require it, so there's no need for debating. Gluconeogenesis is so exhilarating. Glucose, glucose, come to be. Glucose, glucose, come to be. Oxaloacetate has got to turn to PEP, employing energy that comes from breaking GTP. From there it goes to make a couple phosphoglycerates, exploiting enolase and mutase catalytic traits. Oh, gluconeogenesis is liver specialty, producing sugar for the body most admirably. 6 ATPs per glucose is the needed energy. Gluconeogenesis is liver specialty. Oh, glucose, glucose, joy to me. Glucose, glucose, joy to me. Converting phosphoglycerate to 1,3-BPG requires a phosphate that includes ATP energy. Reduction with electrons gives us all an NAD, and G3Ps isomerize to make DHAP. Oh, gluconeogenesis is anabolic bliss, reversing 7 mechanisms of glycolysis. To do well on the final, students have to learn all this. Gluconeogenesis is anabolic bliss. Oh, glucose, glucose factory. Glucose, glucose factory. Aldolase reaction puts together pieces so a fructose molecule is made with 2 phosphates in tow, and one of these gets cleaved off by a fructose phosphatase unless F26BP's acting, blocking pathways. Oh, gluconeogenesis, a pathway to revere. It makes a ton of glucose when it kicks into high gear. The cell's a masterminding metabolic engineer. Gluconeogenesis, a pathway to revere. Oh, glucose, glucose, jubilee. Glucose, glucose, jubilee. From F6P to G6P, that is the final phase. The enzyme catalyzing it is an isomerase. Then G6P drops phosphate and a glucose it becomes inside the tiny endoplasmic reticulums. Oh, gluconeogenesis is not so very hard. I know that on the final we will not be caught off guard, because our professor lets us use the filled out index card. Gluconeogenesis is not so very hard. Yes. Thank you, guys.